If you are looking for brief explanation of biology chapters, then this video is for you. Hello everyone, welcome to the Biology Funda. I am Prakina, your biology educator, and today we are going to discuss Class 12 Biology chapter Sexual Reproduction in Flowering Plants. First, let us learn about the definitions. There are some definitions mentioned in NCERT textbook. Some of them are mentioned above. These are very important. First is Microsporogenesis. Now, what is Microsporogenesis? Micro means the word comes from microspores. Genesis means synthesis. So, it's a formation of microspores from a pollen mother cell through meiosis. What is Megasporogenesis? Megasporogenesis, the word megasporo comes from megaspores. Genesis is synthesis, so it's a formation of megaspores from the megaspore mother cell. Next we have pollination, which is a mechanism of transfer of pollen grain to the stigma of the pistil. So these pollen grain are shared from the anther. Then we have double fertilization. This is a unique event that takes place in flowering plants, which are angiosperm, and this involves two types of fusion. One is syngamy and the next one is triple fusion. And this fertilization process, double fertilization process, takes place in embryosphere. Then we have apomixis. This is a mechanism to produce seeds without fertilization. It's a form of asexual reproduction that mimics the sexual reproduction. Then we have polyembryony. It's the occurrence of more than one embryo in a single seed. Then we have parthenocarpy. This is a development of fruits without fertilization. When I say without fertilization, that means seeds won't be produced in the fruit. They are seedless. Example is banana. Then we have perisperm. It's a residual position nucellus found in some seed like black pepper and beet. Next we will learn about the male reproductive unit. Male reproductive unit is androecium in flowers, which consists of whorls of stamen. Now if I draw a stamen, it looks like this. This part is known as the anther, which is at the terminal, and this long stalk is called filament, which is attached to the thalamus or petal. Anther, if I cross sectioning, you will find the structure like this. This anther. There are two lobes. That's why it is called as bilobe. In each lobe you will find two theca. So there are four theca actually, but since in each lobe there are two theca, it is known as dithecous. In this theca you will find microsporangia. This is a tetragonal structure. This microsporangia, if I draw like this, you will find that there are four layers outside. These four wall layers are endodermis, endothecium, middle layer, and tapetum. Epidermis. There are four layers: epidermis, endothecium, middle layer, and tapetum. Epidermis is the outermost layer, then in the middle there is endothecium, then middle layers, and lastly the innermost layer is the tapetum. These provide protection and aids in dehiscence to release the pollen grain. And the last layer nourishes the developing pollen grain and it possesses a dense cytoplasm. The pollen grains are formed here in this part. There are two layers of pollen grain. The outer layer is known as exine, so external exine. Internal is intine, which is thin inner wall. Exine is made up of sporopollenin. Intine is made up of cellulose and pectin. Sporopollenin is the most resistant organic material which can resist any acid, any alkali, any dangerous vector. Now in the pollen grain you will find cells. The large cell is known as vegetative cell. Then there is another cell which is generative cell, which is small and it floats in the cytoplasm. The vegetative cell is for the food reserve. Then we have female reproductive unit, which is gynoecium. Now if I draw the female reproductive organ, which is pistil, it looks like this. Now gynoecium consists of the wholes of the pistil or carpally. The first part is stigma, which is here. This is the landing platform. This elongated part is the style, and this basal bulge part is known as the ovary. In the ovary you will find ovaries, which are Megasporogenesis, which is attached to the placenta by a funicle. There is a junction between ovule and funicle, which is known as helium. There are a protective envelope around this ovule, which are known as integuments, which has a small opening called as micropyle. And chalaza is the basal part of the ovule. The embryo sac, the mature embryo sac consists of 8 nuclei and 7 cells. There are 7 cells. Let us learn about it. There are egg apparatus, which consists of 2 synergists and 1 egg cell at the micropylar end. There are antipodal, which are 3 cells at the chalazal end, and in the middle there is a central cell which consists of 2 polar nuclei. If you calculate it, you will find that there are 7 cells and 8 nucleus. So that's why it is known as 8 nucleate and 7 cells. Let's learn about pollination. Pollination, there are different types of pollination. One is autogamy, that means in the same flower, these flowers are bisexual, where you will find both male and female reproductive organs in one flower. Then we have geitonogamy, which means different flower but of the same plant. They are genetically similar to the autogamy. And then we have xenogamy, which means different plants, and these can bring genetic variation. Now agents of pollination are abiotic, that means non-living, like wind and water, and there are biotic, which are animal and insect. Now there are some outbreeding devices, mechanism to discourage self-pollination and prevent the inbreeding depression, such as non-synchronized pollen release, stigma receptivity, self-incompatibility. Then we have double fertilization. Now this is a event that takes place in flowering plant. There are 2 fertilization. One fertilization is when male gamete fuses with egg cell to form zygote, which is diploid. There is second fertilization when the second male gamete fuses with these 2 polar nuclei to form primary endosperm nucleus, which is triploid. The first fertilization is known as syngamy. Second fertilization is known as triple fusion. Zygote later forms embryo. Endosperm later forms primary endosperm cell. Primary endosperm nucleus then forms the primary endosperm cell and then forms endosperm. This endosperm is helpful after the fertilization to the embryo. So let's learn about the post-fertilization event. Endosperm, it nourishes this embryo. So it forms earlier than the embryo. There are two types of seeds, non-albuminous and albuminous. Non-albuminous means there is no residual endosperm like in pea and groundnut. Albuminous means retain some part of endosperm like wheat, maize and castor. Fruit develops from the ovary. This ovary wall develops into the pericarp, which is the outer layer. Now there are two types of fruit, true fruits and false fruit. True fruits is when they develop only from the ovary, and false fruit are developed from the other part like thalamus. Like example is apple, strawberry. Now that's it. Also I want to tell you that fruit develops from ovary, and ovules which are found in ovary develop into seed. So this is the end. Thank you so much. If you like this video, please subscribe and share with your friends. I try to cover up more such chapters in a very briefly order that can help you for NEET and boards. Thank you so much. देव बरे करू.